So welcome everyone, and today we will going to be going through the enzymes mega tests. So let's dive in. So here is enzymes. Let's expand it. Expand enzymes revision. Expand it. And here we have enzymes mega test part 1. Let's open it. Now, there are how many questions? This mega test includes 28 enzyme questions. Total time for this mega test is 28 minutes. You have 1 minute for each question. Okay, let's get started. Start quiz. Now, you can see here that we have the timer started. So 27 minutes remaining. Okay. Question 1. Which statement about enzymes is correct? Enzymes become the part of the product. Lowering the pH always slows down the reaction rate. Raising the temperature always increases the reaction rate. So the specificity of an enzyme depends on the shape of the active sites. So we need to select the correct option. So I think this last option is the correct one because it states the exact or right statement for enzymes. Now let's see. Specificity of the enzyme. Why the enzyme is specific? It depends on the shape of the active sites. Right. This is correct. Check. Got it right. Next, the diagram shows a protease enzyme catalyzing the breaking part of the protein molecule into smaller pieces. Which diagram has 3 correct labels? So in this first A, we have the product. It's labeled. So this is substrate and it's a product as well. But enzyme is this one. And here it's saying these are the active sites of enzymes, and this is the product, this is the enzyme. In C, it's saying this is the substrate, this is active sites, and this is enzyme. And here in D, it's showing the active site, substrate, and product. So here the product is not this because it's broken down. So I think these are the products. The enzyme is labeled correctly and the substrate, but it's saying the same thing, the substrate, as a product. So that's not possible. And B, the product is not that. The product is formed after the enzyme have worked. So this is a substrate. So that's not correct. Now in C, this is substrate. This is active site of the enzyme, which is true. And this is enzyme. That's perfectly right. Now let's go on. Check it. Got it right. Next. Okay. Now, what controls the speed of the chemical reactions in all living cells? What controls the speed? Enzymes. Next, the diagram represents the shape of the enzyme molecule, of an enzyme molecule. Okay, this is an enzyme. Okay, with which substrate would this enzyme most easily form an enzyme substrate complex? So which of the substrate can exactly fit in to this enzyme? So this cannot fit in. This even cannot fit in this smaller piece. These are the active sites. This is too big to fit in, and this one can perfectly fit in. As you can see, this part can exactly fit in to this part of the active sites of the enzyme. So I think D is the correct option. Check it. It's correct. Now next, the graph shows how the activity of enzymes varies with temperature. What is the best or optimum temperature for this enzyme, and at what temperature is enzyme not working? Okay, what is the best temperature? How can you get to know about it? We can just see where there is the highest rate of enzyme activity, means there it is working the best. So we can just move down and see what's the temperature here. It's 50 degrees Celsius. So on this 50 degrees Celsius, the enzyme activity is the best, the highest. So in the first column, we have the answer 50. So from these both C, or it could be C or D. From not working, so at 60, we can see at 60 degrees Celsius, the enzyme activity is 0, means it's not working. So it's 60. Now 50 and 60. So it's D. We're done with it. Next, the graph shows effect of the temperature on the action of an enzyme. Why does the rate of reaction change when the temperature is increased from 20 degrees Celsius to 30 degrees Celsius? Now, it's asking why there is the increase in the rate of reaction as the temperature is increased. Sorry, rate of activity of the enzymes. Okay, more kinetic energy of particles. Yes, that's true. Because as the temperature increases, the kinetic energy of particles increases and they collide more frequently. So means there should be the tick or correct. So A or C could be the correct option. Now, more frequent collisions of particles. That's true. So A is the correct option. Got it correct. Next. Question 11. The graph shows how the enzyme controlled reaction is affected by the temperature. So the temperature versus the rate. Okay, what statement explains the change in the activity between X and Y? We can clearly see that the rate of reaction is decreasing. Okay, the kinetic energy of molecules has increased. Okay, there are more effective collisions. There is a change in the enzyme shape. There is more substrate present. Okay, as the temperature is increasing, the enzyme's activity or the rate of activity is decreasing. It is due to the denaturation of an enzyme. And denaturation is the change in the enzyme's shape due to the high temperature. So this is the correct option. And we got it correct. Next. Now an experiment was carried out to investigate the effect of pH on enzyme action. The graph shows the result, and y-axis and x-axis. This vertical axis is known as y-axis, and this horizontal axis is known as x-axis. What are the labels of the x-axis and the y-axis? So we were already studying that with the graph for the temperature versus the activity of the enzyme, rate of activity are on the rate of activity is on y-axis, and the temperature so in this case the pH will be on our x-axis. So x-axis will gonna be pH, and the y-axis will gonna be rate of reaction. So this is A. Check it. We got it right, I think. Yes. Okay, next. The graph shows the activity of 3 digestive enzymes at different pH levels. Which statement is correct? Okay. The enzyme X and Y are both active at pH 7. So pH 7, they are active. This one, that one is active, increasing, and Y is okay. X and Y are both active at pH 7. Enzymes Y and Z are both active at 8 pH 8. Y and Z at 8. Wait, 8. They are both active. You can see this one is active and this one is also active. And the first was not wrong. The first was not correct. It was wrong because I was confusing Y and Z with this X and Y. It seems like this one is correct, but let me go through the other ones. Enzyme X and Z are both active at pH 4. X and Y pH 4, no, very less active. This one's active. Okay, enzyme X and Z. Okay, enzymes Y Z are both active at pH 4. Y Z are active 4. No, so this one is correct option. Let's go through it. It's slow here. It's correct. Now next. And now, what is the correct definition of the term enzyme? So what exactly is the correct definition for the terms enzyme? Right. Okay. The enzymes are proteins that act as biological catalysts. That's right. We just studied in the previous video, if you remember, in the slides. This one is correct. Now, next. Okay, the diagram shows the difference between the Diagram shows the activity of the salivary amylase, pancreatic lipase, and stomach protease at different pH levels. So from the graph, what is the optimum pH for the protease enzyme? Okay, so in stomach, we know in stomach the pH is the medium is acidic, not alkaline or neutral. It's acidic. So acidic is below 7. So means it should work perfect before 7, below 7 pH. So we can clearly see this one could be the correct. Here it's the amylase, the protease. So protease is working well at 2. You can see that this is working at 2. They're perfectly working at 2 pH. Right. The graph shows the effect of pH on the activity of 3 different enzymes. The table shows the pH in different parts of the alimentary canal. Okay. It's saying the part of the alimentary canal: mouth, stomach, small intestine. Okay, pH 7, 2, and 8.5. Which enzymes in the graph are likely to be protease enzyme? We just studied that in the previous question. We just revised the concept that in the stomach the pH is acidic. So means that where the enzyme protease is present, we just saw it was in stomach. So it was in stomach. So what was the pH for that protease enzyme? It was 2. We can see it from the graph as well. The X is their protease. So here we can see that this one, that this protease is also present sometimes in the small intestine, where the pH is higher than 7 and it's alkaline medium. So this is shown by Z. So X and Z are the correct option. Check. And we got it right. Now next. Question 17. The experiment was carried out to study the effect of temperature on the time taken for protein to be digested by an enzyme. The table shows the result. We are given with the temperatures in one column, and in second column we are given with the observations. So what we want to do is, for these results, at which temperature does the enzyme denature? So we know the enzyme denature at the highest or the maximum temperature. So we can see that here the maximum temperature given is 55 degrees Celsius. And the observation is also given: no digestion take place, as the enzyme has denatured and its active site has completely destroyed. So 55 is the correct means somewhere near 55. So here, when the answer, or maximum we are given with, is 50 degrees Celsius. So we're gonna select this one. Check it. That's right. Next. Okay, which statement describes the effect of temperature on enzymes? High temperatures denature enzymes, making it difficult for substrate molecules to fit into active sites. That's right. High temperatures denature enzymes, making them easy substrate. No. Let's check it. That's right. Next. Which graph shows the effect of temperature on the activity of enzyme? Okay, of an enzyme. Okay. So we know as the temperature is 0, the enzyme activity is too slow. So at a body temperature, which is approximately at 37 degrees Celsius, the amount or the rate of the activity of enzyme is the highest, and then as the gradually the temperature is increasing, the enzyme get to denature and the rate of activity decreases. So A could be the correct option. Now next, the graph shows the effect of temperature on an enzyme control reaction. Okay, so the rate of reaction and temperature are given on the graph. Which statement describes the effect of temperature on this reaction? As you know, as the temperature increases to 25 degrees Celsius, the reaction speeds up. That's right. As the reaction is at the temperature 25 degrees, approximately here, the 25 degrees Celsius could be here somewhere. There, the reaction speed step, that's right. Between 10 degrees Celsius, 20 the enzyme stops working. No, it was not stopped working, it was increasing. The optimum temperature for this reaction is 40 degrees Celsius. No, it's not right. It's 40 somewhere here. So it should be somewhere over here. So that's not right. Okay. Between 35 degrees Celsius and 45 degrees Celsius, the enzyme have stopped working. So 35 to 45. It's 45 here, something or somewhere over here. So we know between 40 and 60 it's 50. So 45 is here. That's not right. So I think this is the correct option. That's right. Next. Now 21. We're left with a very less time. Okay. Many enzymes do not work at temperature above 60 degrees Celsius, which this man explained this. Okay. Product molecules are not made because of active sites change shape. Yes, they denature. Right. Oh, very less time remaining, and we just left with 7 questions. Okay, 6. The diagram shows the, and we have already discussed. So Q is the substrate. Q from these both could be the option. P is the active site. Active site is P, so it means this one is the option. R is the product, and S is the enzyme. D. Check it. That's right. Next. And now we are in the diagram shows an enzyme with the substrate product molecules. So we know the Q is the substrate which is being fitted in the enzyme, which is P, and then the products are formed. So which form the enzyme substrate complex? So Q and P and Q form enzyme substrate complex. P and Q. That's right. Now next, which group of we have very less time remaining. Okay, which group of compounds ensure the metabolic reactions take place effectively? Okay, enzymes. Next, enzyme acts as digest protein in the stomach. 4 test tubes were, and I think we have done with the protein and enzyme. The test tubes are kept. Okay, in which test tube the protein digestion be quick? So the pH should be very low, because it should be the acidic medium in the stomach. So 2, and the temperature could be high as 35, which is approximately the body temperature. So B is the correct option. Let's move fast. And now we are here with the diagram should represent the action. Okay, where the X remains the unchanged, so it's an enzyme, and these W was the product, and Y is the W was the substrate, and XW, and Y was the product. So C is the correct option. And now the last, and we have the very last time remaining. Next, oops, time ended. I left with the last question. Okay, no problem. What score did I got? It's 82.14%. Just because of you guys. Okay, so we are done with it. So thanks, take care, and see you in the next video.